come and be part of, okay, recording started. She was going to be part of our SASSB conference, which was going to happen last year. And so we're very pleased that she's joining us, at least virtually today. She's a senior research scientist at the Spanish National Research Council in Madrid at the Botanical Gardens there. And she describes herself as an evolutionary biologist, and she's interested in the theory and methods of biogeographical inference. And her research focuses on the analysis of, to date, mostly macroevolutionary patterns of distributions across a diverse array of organisms, and the development of new analytical tools, and especially Bayesian inferential tools. And after her PhD from Spain, she moved to Sweden and worked with Professor Fredrik Ronquist there in several different jobs. And their collaborations pioneered the development of statistical approaches in biogeography based on process models. And Dr. Sanmartín has published articles in top-tier cross-disciplinary journals, and these studies have had a profound influence on the field of biogeography, as can be seen from their metrics and their high citations. So her work has over 8,600 citations. It's also been cited in classic textbooks and popular science books. And she's been invited as a keynote and plenary speaker at more than 30 international meetings, and regularly lectures workshops in Europe and South America. And so tomorrow, we're very pleased that she's going to be adding South Africa to that list. So I'm very pleased to introduce Isabel, and I think I'll leave you to give your title. So over to you.
Yes, thank you. So I guess I need to start sharing my screen now. And yes, first, I'd like to thank Nicola and Fanus and Antoine and then all the people at the South African Systematic Association for inviting me to be here. We organized this 1 year ago, but the pandemic didn't allow.
Also, because you had some unexpected field trips.
Physically, so so I'm very happy to be able to give the seminar at least virtually. So I will do that, share my screen. Let's see. Hi. Okay, so I think it's right now. I'm actually going to keep the like this, because if I go into full mode, maybe we'll stop. Is it too small now? Or you
can continue like that.
Okay. So the title of my talk today is the Rand Flora Revisited, and I'm going to discuss this floristic pattern, an African Mediterranean pattern that also affects Canary Islands, and I'm going to do it in the context of presenting new methods, new approaches for reaching the micro and the macroevolutionary levels in biogeography and evolution, which is a theme that has interested me and my team for a long time. And you will see why this pattern is so appropriate for the particular subject we have today. Oh, what? I know what. Let's see if let's try with this, because now it doesn't. Okay, now it works. So we all know that biodiversity is not evenly distributed on Earth. For example, it's not distributed equally across taxa. I work with two different genera of angiosperms, flowering plants. One of them is Hypericum, St. John's wort, and it includes around 500 species, even more now. And I also have a genus that is distributed in Africa, in the Canary Islands, with only 3 species, which is called Canarina, belonging to Campanulaceae. But biodiversity is also not equally distributed in space over the globe. So the slide on the right represents the famous latitudinal diversity gradient, the discovery that species richness, diversity is larger close to the equator, and it goes down, diminishes close to the poles. So for a long time, that pattern was explained in terms of environmental factors. For example, temperature and precipitation, that's shown on the slide. So the idea is that those environmental factors, differences in temperature, precipitation, evapotranspiration, and will, in a way, explain those patterns. But it was very clear when, in the mid-20th century and later, with the appearance of molecular systematics, that the pattern can also be explained by evolutionary processes, such as, for example, dispersal, extinction, and speciation. And that's the idea on this slide. So those environmental factors, they are important for because they sort of act or affect these evolutionary processes. So speciation, extinction, and dispersal rates, they are not regulated, but they are influenced by ecological factors. So at the end, it's an interplay between ecology and evolution, acting through time and over space. But there are also two other dimensions that are important on this problem. One of them is the idea of evolutionary dimension. So here on this slide, I show what I call macroevolution. In macroevolution, we try to explain patterns that occur at the species level and above. So genera, and lineage, close related species, families. And we do so by reconstructing phylogenetic relationships with morphological characters, mostly in the past. That's what I did for my thesis. And now we mostly do with molecular characters, with DNA sequences. And with those DNA sequences, and we have a molecular clock, we can be able even to reconstruct how those species build up over space and over time. So we can build a time tree showing how the species diverge from their closest relatives, and then how was the rate of diversification in the group, what was the rate of extinction. And we can also take the more spatial approach, and that's actually the focus of most of my research, biogeography. So we want to understand why species are where they are, how did they get there, when did they get there, and we can use geographic data in the present, sometimes fossil data, but you also move into the field of ecology because we can use this expanding field of ecological niche modeling to understand what are the environmental factors explaining why they live where they live. So what are the climatic tolerances, what is the range of temperatures they can live with, or what is the limit after which they die. That would be the realm of macroecology or ecology. But recently there has been a more closer focus within the species. So the infraspecies level, the population level. And why is that? Because it has become very clear that even if the patterns are often explained at this macroevolutionary approach, so at this large approach, the microevolutionary patterns or process, those that explain genealogical relationships, and they are very important because they are at this interface between the population and the species. So they can explain things like why a species was formed, which were the actual factors that led to the population divergence, division. And then we need to think in terms of, I said, genealogical relationships. So, for example, we have this coalescence-based theory that explains how populations, how are they connected in the past, and which were the factors explaining that. And we usually work with data that is molecular data that is more variable, like haplotype networks. We have also used fragments of DNA, FLPs, recently with single nucleotide polymorphisms. And at the biogeographic level, we are not so much interested in where did the species disperse, when, but more into the population level approach. That is, what is the, how fast the population expanded. And then we think in terms of a continuum space, like in the right part of the slide. So we move into phylogeography. So we try to find which was the location of the ancestral population before it became so widespread across this area. And we move into landscape genetics, and we're thinking in terms of coalescence theory, as I said, so population bottlenecks, geographic expansion, the rate of geographic expansion, the rate of how alleles expand.
This is
Okay? Can you hear me?
Sorry, Isabel, I think there's someone who's not muted their microphone. Could
Okay,
everyone just
right.
double check? Sorry, we've been having it off and on throughout the talk. Sorry, Isabel. Everyone
It's
can
okay.
just check.
Yes, no problem at all. I was just thinking that sometimes I switch off and then it's like, I don't know whether you are there.
No, we're all
That's
listening.
okay.
We can hear you.
Okay. So there is one problem with that. And there is, of course, there is no clear separation between the macro and the microevolution. So there is all a continuum. So this slide tries to show that. I mean, you can see on the right part that we start with population genetics. We start with one single population. Maybe there is some subpopulation structuring. But then over very long time periods, and I'm thinking in terms of thousands and even millions of years, then those movement of individuals within populations that are the focus of microevolution, landscape genetics, the movement of alleles, how they became dominant or rare or why, how are they haplotype network structuring, then those movements and those events that are studied over a continuum, landscape, two-dimensional space, they become saturated, similar as when you have the problem with third codon positions in a protein. So it's very difficult to infer them because there have been so many, so repetitive, that it's very difficult to infer them. So that's when we move into biogeography. Then we are interested in the rare events, the events of moving between continents or moving across a geographic barrier, moving into a closed region. So the rare events of migration of a species that changes its distribution and colonizes a new area, those would be the equivalent to the rare events of mutation, for example, in the first and second codon position. They are more easily detected at the macroevolutionary level because they are rare, they are not repetitive, they are not saturated at the species level. And that's where population level biogeography or phylogeography, they dissolves into biogeography. But the problem is that often we use different methods, we use different data sets, we use different assumptions, frameworks, analytical, and that becomes very complicated. So just going to the subject of my talk, I'm going to show ways to escape that paradox or to combine both using the Rand Flora pattern, or at least that's what we're trying to do. Not that we have achieved to do that, but we're trying to do. So what is the Rand Flora? The Rand Flora was described in the 19th century by a Dutch botanist, and it describes this pattern that is represented in red. So it's the idea that there are sister taxa, sister species, sister subgenera, and sister close related species, clades, that are distributed on the borders of the African continent. So on the edge of the African continent, because Rand Flora is like the edge flora. And those are represented here on these red blobs. So they are present, for example, in the Eastern African mountains, they are present in the Ethiopia and Somalia and on the high plains. They are also present in South Africa and in Northwest Africa, in Cameroon, and they are present in their close by archipelagos. So for example, in Socotra Archipelago and in Canary Islands. And my interest on the pattern came because I was working on the Canary Islands, and many of my plants, they have sister groups in Ethiopia and in Oman and Yemen, so to the other side of the Sahara. And so the question was why, how can it be? How did it come to be separated by thousands of kilometers of a desert or even a tropical Central Africa region? And the other interesting question is that it's not a pattern of one genus. It affects many families of angiosperms. So it's present in Asteraceae, Campanulaceae, Plantaginaceae, for example, very different Scrofulariaceae, very different angiosperm families which evolve at different times and which have different means of dispersal and of seed dispersal and pollination. So what is the explanation for the Rand Flora? The main idea is that, or the classical idea is the Rand Flora is explained by either dispersal or extinction. So one possibility is that the pattern originated, the ancestor of these groups originated in one region, for example, in the Mediterranean or in Canary Islands, and then it moved into Africa and then dispersed to the other isolated areas and stayed there and diversified there. And it could come from the north or it could come from South Africa, when the hypothesis is that, and then Southern Africa, and then move north. The other idea is that it's a pattern explained by vicariance, by extinction. These are groups that were widely distributed in the past, and they experience extinction. So now they are climatic relics, refugia, living in these refugia at the borders of the continent, because those regions are more climatically suitable for them. So there are certain evidence about the Rand Flora vicariance extinction hypothesis. One of them is that, of course, the African climate has changed through time. We know from plate tectonics that Africa moved from the supercontinent of Gondwana in the Mesozoic around 100 million years ago and started moving north eastwards and collided with Eurasia. And that process of moving into the equator zone and collision and uplift of the east of the African mountains on the east, it started an aridification trend. So some regions became very arid and at least moved from tropicals to subtropical, and then even to arid or semi-arid, and that's represented on this slide. We also know from geology and from climatologists that at least in the early Miocene, Africa was very different to what is today. So there was a desert in the central of Africa, close where Congo is now. It was called Miocene Congo Desert, a bit to the south. We know that from rocks. And we know that the Northern Africa was much more tropical and the vegetation was not what it looks today. And then as the climate became, as I said, more dry and xeric, the north became more arid and the central Africa became more tropical. So it was called an aridification versus tropicalization process. And that's a combination of different geological tectonic events. But that major change in climate and in, of course, in physiognomy of vegetation, it has profound implications on the vegetation of Africa and also on the evolutionary fate of those lineages. And the Rand Flora is interesting in macro and micro because there are many different evolutionary levels. And some levels affect genera and others affect species complexes, even species, and even within species, there are some examples. So it's different, probably different times at which the Rand Flora was formed. We've been working on the Rand Flora for a long time now, maybe around 10 years, and we've been slowly working on different genera, approaching different genera. These examples here are based on Sanger sequencing of chloroplast and nuclear markers, ITS, and on the left, you can see Plocama, that's a Rubiaceae, and it has this famous pattern in which there is one species in the Canary Islands, a group of species in Southern Africa, and then there are species also in this is a bit there are species even in the Southern Arabia, and they reach even the Middle East and the Iranian plateau and everything. But there's one clade that shows clearly a Rand Flora pattern formed by Plocama pendula, for example, and Plocama yemenensis or Plocama crocilis. And on the right, you can see Euphorbia balsamifera. That's a famous pattern because it's only one species, Euphorbia balsamifera, which is now 3 species, but in the past was formed by 3 subspecies, one in the Canary Islands, one in the Sahelian region, Mauritania, and one is distributed adenensis, expressing only in Somalia and in Southern Arabia. So what's the problem we face when we want to study the Rand Flora pattern? One of them is that we're dealing with extinction, and extinction is very difficult to measure. So in this slide here on the top, you can see that why extinction is so difficult for us. People, scientists working with neontological data, with present day species, live species. And you can reconstruct the phylogeny of the group. For example, the true phylogeny could be the one on the left. And then we have both extinct and extant species, taxa. But then if we move to the fossil record, we only have the extinct. So we have like a part of the phylogeny, but mostly we have a time tree, a phylogeny with molecular data. So we only have that phylogeny on the right. So you can clearly see that both the fossil record and the time tree, they only reflect part of the pattern. So we need to come with ways to infer extinction from species that are alive today, but they share phylogenetic relationships, and those are evidenced by DNA sequences, by shared DNA sequences or mutations. So what we do is, I'm not going to go into detail, what we do is that we use the time tree, the molecular phylogeny, we use molecular clocks, we have dating the phylogeny, and then we use mathematical models to infer the rate of speciation and the rate of extinction. And the rate of extinction is inferred indirectly. So the rate of speciation is the rate of origination, but the rate of extinction is inferred, if the process is constant in time, is inferred by the change in the shape of the phylogeny, if there was no extinction versus if there is extinction. So that's called the pool of the present. And that's done, here is shown using these time plots, like lead through time plots, like curves, but now we are using more modern or more sophisticated methods using maximum likelihoods and Bayesian methods to infer the rate of speciation and the rate of extinction. But there are also another problem, is there are two types of extinction. One of them is called, on the left, is called the background extinction. That's the rate of extinction that is intrinsic to the clade. So that's something, is at least the paleontologists see that the idea that extinction that is associated to some particular traits of the clade. So it's extinction that is constant over time, over the history of the clade. Sometimes we call the red queen extinction, the background extinction. But on the right is what is called the mass extinction. And it's sometimes represented by this Court-Jester model. The idea is the mass extinction is not intrinsic to the clade. It's something that is outside the clade. It's an extinction event that affects many clades simultaneously. So it doesn't depend on particular characteristics of a lineage that is more adaptive or is more prone to extinction. It's, for example, the effect of a meteorite colliding with Earth, and then everyone that is living at that time just dies out. So mass extinction is usually represented on this slide an event that at some time point it removes part of the standing diversity. It's like for example, 90% of all living taxa at that particular time t just disappear and die out. That's very different to background extinction. That is a more constant process. For every 2 species that appear, 1 gets extinct. So why is it that difficult to disentangle? There are many different explanations for that. Some are mathematical, they have to do with parameter and identifiability. We have models that have parameters that are non-identifiable from other parameters. But there is also this idea I want to show in this slide, and is that mostly we often represent mass extinction as a single pulse event, something that happens at time t, instantaneous. That makes sense, for example, for not even for a meteorite colliding. But the paleontologists criticize this model we neontologists or biologists working with living taxa use, because they say that mass extinction, it takes place during an interval of time. Usually they think in between 1 million year, 5 million year geological time, everything, a percentage dies out. And that's reflect on these slides here. So you have a slide in which we represent this mass extinction as one event in time, and that's inferred by this parameter rho. And another is an interval time when extinction is larger than speciation. So it's a period of time very short, because otherwise the clade will die out, in which extinction is larger than speciation. And that's very interesting for the Rand Flora, because we think that the Rand Flora was formed over time in interval times when extinction was larger than speciation, and then diversity recovered. So going to the actual patterns. So what we did is that we collected as much as we could reconstructing genera and the phylogenetics of these genera. We use molecular data, usually DNA sequences. And for example, this slide shows many different genera that shows the Rand pattern. And we went and built the phylogenies, and we also dated those phylogenies with molecular clocks. And then we went to see what we could get from those phylogenies. One thing was clear is that most of these junctions, those east-west sister taxa, they seem to diverge during a period that is called the late Miocene and early Pliocene and late mid-Pliocene. So the Miocene and Pliocene were periods of major climate change in Africa, and they were affected by periods of very dry climate. And the Rand Flora seems to support that idea that it was formed during those periods. but it was not formed in one particular point in time. It was formed at several different times, but all within that period that is represented on the slide with the red square. Another thing we observed is that the Rand Flora taxa, when we estimated using our models for extinction, when we estimated the rate of diversification, the ratio of extinction to speciation and different speciation extinction, we could observe that the Rand Flora genera have very low diversification rates, pointing to extinction. And that's on the right part of the slide, where we compare the Rand Flora genera against their families they belong to, or tribes. We could see that they were outsiders, outliers in those diversification patterns. They were maybe poorer, species poor, because they have very low diversification rates. And we also use these more sophisticated methods, Vagisian methods, to infer periods in which they actually experience high extinction rates. And that's shown here for Plocama, that genus I mentioned in Rubiaceae, and with species in the Canary Islands and Eastern Africa, Southern Arabia, and Southern Africa. And we saw that actually this genus experienced a period of very high extinction in which extinction was higher than speciation during until 10 million years ago. That's between 20 and 10 million years ago, because then the diversification was negative. Extinction was higher than speciation. But we can also, I mentioned that we are not only concerned with time, we are concerned also with space. So I'm a biogeographer, so I'm interested on spatial patterns. And extinction can also be studied from a biographic point of view. So not only the rate of extinction, but where did extinction take place? And those where these biographic methods come about. For example, the methods I use are called model-based or parametric methods in biography. We use models called Markov chain Monte Carlo models that are also used in phylogenetics. And we try to infer the rate of extinction per lineage and also per area. And as well the rate of dispersal, which would be like the counterpart of extinction. And in this slide, don't want to go into detail because I think I'm taking already too long, but just to say that there are models in which we estimate parameters and they are based on molecular phylogenies with time trees, with a temporal dimension. And we also, as I said, we are interested in where extinction took place. So we use these more geographic dependent extinction methods. So what we got? So what we got is that first, we were able, of course, to infer, this is an example from on the left for Plocama, this genus I mentioned before, and on the right is Cantoloma, a genus with only 3 species, one in Namibia, one in Somalia, Southern Arabia, and one in the Canary Islands. And we use these biogeographic model-based methods to infer the rate of extinction, local extinction, and the rate of dispersal. And we saw the rate of extinction was high compared with the rate of dispersal for these groups. And we also saw that the ancestors were more widely distributed in the past, and now they are more reduced distributions. They have a reduced geographic range. So we confirmed that. But we also observed something interesting. When we compared the rate of extinction, for example, in genus Hypericum, I mentioned in the first slide, St. John's wort. So 500 species, cosmopolitan, everywhere except Antarctica. And what we observe is that this genus has a higher rate of extinction in Africa, in the African clades, than in any other geographic regions. And that's shown on the slide here. So the relative extinction, the rate of extinction is higher in the African taxa, in red, on the left phylogeny, than in the rest of geographic regions that are present, for example, North America, Europe, Australia, South America, etc. And the thing is, of course, I mentioned that the Rand Flora affects many different families of angiosperms. And I've been discussing so far only one genus at a time. So I did independent analysis. But what about if we want to do multi-clade analysis? We want to infer extinction at the biotic level, at the flora level, Rand Flora level in this case. So we can use these models that are called Vagisian Heraldical models. And tomorrow I will show a bit about that. So these models, they not focus only on one particular genus, but we want to infer extinction across genera. So the models are built in a way that you have different data sets, molecular phylogenies or DNA sequences, and you have different molecular clocks, so the groups are not the same age, but you are still able to infer the rate of movement between areas and other parameters that I call carrying capacities that are equivalent to the diversification in situ. And with that, you can partition to see the role of extinction, and you can do that across clades. So this is an example for the Rand Flora that I published some time ago, and we are actually repeating the analysis with our new genomic datasets. So what is shown here on the left slide, you can see this the Rand Flora. So that would be the yellow blobs, well, the lines in red marking the Rand Flora regions. For example, Southern Africa, Eastern Africa, Southern Arabia, Canary Islands. And we could see is that the dispersal between these areas was very high in the past. And it's surprisingly high between, for example, Canary Islands and Eastern Africa, Southern Arabia, even though now none of these species live on the Sahara Desert. But we could infer the rate of extinction, historical dispersal, in that time. We also saw interchange or dispersal between Southern Africa and Eastern Africa. It was not as common, but it's still very common. And of course, with Mediterranean and after the collision with West Asia. But it was also very interesting to see these yellow blobs, because yellow blobs indicate how much in situ diversification there has been in those areas. And you can see that Southern Africa comes a very large yellow blob, yellow cycle, and the Canary Islands is that tiny on the left yellow blob. So it shows, like, confirms the idea the Canary Islands was formed not so much by in situ diversification, but by exchange with other regions. So those arrows going to the Canary Islands, they are very large. Whereas Southern Africa is more in situ diversification and little on exchange. Of course, that's for the Rand Flora, not for the angiosperm flora of Southern Africa. And that could be explained by the climatic and geological history I mentioned before, because we know that desertification, aridification start in the south and then proceed to the north. So one of the last parts of Africa to become arid was Northern Africa. I'm going time, oh my, I think I'm going a bit slow now, but I will try to not go over time. So one of the first slides I mentioned that it was important to consider environment. I mean, we're dealing with extinction, dispersal, speciation, but we also need to think on the environment. I mean, the factors that are somehow regulating or have an influence of those rates, evolutionary rates. For example, temperature, precipitation, as I say, productivity, evapotranspiration. And one idea that is very interesting is the idea that you can have ecological vicariance, that is one population that is divided into two disjunct populations, not by a geological barrier like a mountain range or a desert like the Sahara, but by an ecological barrier. Suddenly the climate changes inside the population, inside the geographic distribution of the population. So the extremes, these populations are the extremes at the end of the distribution become isolated. And the allopatric speciation is not driven by geology or by geographic barrier, but by an ecological, environmental barrier. The best example is the Sahara. So the formation of the Sahara in the late Miocene, it probably drove some of the disjunctions in the Rand Flora, at least for those that are not the oldest, but that intermediate oldest. So that's the idea of ecological vicariance. But for that, we need to reconstruct the ancestral tolerances of the climatic tolerances of the ancestors. So that's what we did. We use macroevolutionary models that call ecological niche modeling, and we reconstruct the models, the ecology of those Rand Flora genera, and try to know where were the ancestors and what were the climatic tolerance of the ancestors. What we found is that those actually reflect very well the age of the Rand Flora disjunction. So how old is the divergence of the Western Africa and Eastern African taxa? So those genera are more adapted to subtropical or tropical conditions. They have older divergences, Rand Flora disjunctions. And those that are more temperate or semi-arid, xeric, they have younger disjunctions or separation between Eastern Africa and Western African taxa, for example. And that's reflected on this slide. So again, it confirms, but with ecological data, that the Rand flora was not built in one particular time period, and it was not built in one time point because of one event of climate change. It was slowly built because of aridification, and taxa reacted according to their climatic affinities. So things about that, limitations with the Rand flora. We have very little data. We do have phylogenies that have only maybe 3-4 species, 10 species, because the Rand flora are species poor because they have been affected by extinction. So we have problems with our methods with that. And we have very little data, not always, but sometimes in terms of geographic records, because there hasn't been so many studies on them. And so they are not very well represented in online databases, like for example, GBIF. So we have problems with that. So I just want to give you a snapshot of what we can do in that case. So we cannot really use these methods, sophisticated methods I mentioned before, model-based, livelihood Bayesian, maximum livelihood. But we can use combinations of both. We can use ecological niche modeling, for example, to infer biogeographic models. And we did that recently. And then we were able to infer, for example, the ancestral distributions along branches that have no diversification. So those very long branches, long internodes in the phylogenies, we have no data, and using this methodology with ecological niche modeling, we were able to infer the ancestral distributions. We even went further and we used simulation-based approach. These are called automatic models, automat models. And what we did is we simulate a process that's very similar to the Rand flora, and we simulate speciation, extinction in the landscape, resembling Africa, and we have the conditions in Africa from the early Miocene to the present, and we simulated 1,000 times, and we observed the outcome. Where do my species appear at the simulation process at time 0? Where and how many species do I have in each of the areas? Does it look like the Rand flora, like a ring pattern? And so we compare the simulations with the patterns we observe, and that's the basic of these simulation approaches that are very different to the one I mentioned before. It's simulation based. And we even were able to infer even phylogenies or to implement the phylogenetic part of those models. So finishing. So macroevolution. So far, you've been listening to me about mostly species level phylogenies. But what happens with microevolution? What happens if my phylogeny has only 3 species? And I can even use simulation-based automats, but still I have 3 species. So sometimes I need to go into the population level. I want to see bottlenecks. I want to see events at the population level. I want to have more data. And also Sanger sequencing is very ineffectual for microevolution because I have few loci and very few mutations, so phylogenies have no support. So recently I moved into the realm of genomics. In particular, I use target sequencing to move from 10 loci to 100 to thousands of genes. And with that, I got more resolution in my phylogenies. And I was also able to study population level and interpopulation level relationships. And this is an example with Euphoria balsamifera, the genus the species I mentioned before. And on the left, you can see, and here on the left, you can see the Sanger phylogeny based on ITS and a few chloroplasts. And you see it's not resolved at all. And now you can see on the left, the genomic phylogenies. You can see the difference. There is much more resolution. You can even get data at the population level for studying population level processes such as coalescence, introgression, bottlenecks. And for that, we use these DNA sequences, target sequences, but we also extract single nucleotide polymorphism, so variable sites. And I use these traditional approaches. For example, here is represent phylogenies based on the actual DNA sequences, the population level for a species in the Canary Islands. But we also use Bayesian methods for structuring populations to see things like introgression on the bottom left. So to see it has been admixture between populations, so they are not completely isolated. There are allele introgression, one of them. We can also compare patterns of geographic barriers at the population level. So this is the classical approach we often use with macroevolution. But there are other ways to do macroevolution that resemble more the macroevolutionary analysis. For example, this is called model-based phylogeography. We use here models that were actually inspired by models used in phylogenetics, but are now used in viruses. They have been used, for example, for COVID, for the pandemics. And what we do is that we try to infer the movement of individuals carrying alleles. So we infer movements of individuals within populations, which is called phylogeography. And this is shown, for example, of Cantoloma, this genus with 3 species in South Africa, Canary Islands, and Eastern Africa. And there are different methods. On the slide, I show 2 of them. Sometimes they give different results, but it's much more fine scale and accurate than the methods we use in microevolution and biogeography. And just in the last year, together with a postdoc, we also use different approaches that are simulation-based. So this one here, as I mentioned, is the equivalent of the model-based approaches we use in biogeography that I show, the island biogeography models that I show for the entire Rand Flora. But this could be more equivalent to the simulation-based approaches that I mentioned, the automat models. This is called deep learning or machine learning, and we simulate the scenarios of microevolution, population bottlenecks, expansion, demographic expansion, divergence, coalescence, introgression, migration, and then we compare these simulated outcomes with our patterns, the patterns we observe in our data. And you can use SNP data or other types of data, and it's very powerful. You can simulate very different scenarios because you don't need to define parameter dependency or the livelihood, analytical livelihood of the model. So it's much more powerful for scenarios. And just to finish, we're actually doing that with our Rand Flora to study patterns like this. For example, it has been how was the colonization of the Canary Islands? Has there been more migration to the left or the right? And in which direction? Has there been bottleneck, etc.? And with that, I finish. 15 minutes over time, and just want to show a slide with my team, Rand Flora team, many postdocs and PhD going through the years, and I thank all of them for, without them, this work hadn't been possible. Thank you.
Thanks very much, Isabel, for a great talk and a great overview. I think it's very hard to put all of that into 30 minutes. So hopefully in the question times, people can pull out some of the things that they found most interesting. So I think we'll take some time now for questions. If people would like to ask, please maybe just quickly introduce yourself before you ask your question. So the floor is open.
maybe it was too much for it was like such a barrel of ideas.
Yeah, but at least people can we'll have the recording so people can go over it and pull out the things, and that'll be great. But I've got just a quick question. Actually, when I first came across this idea of the Rand Flora, I think it was your 2010 paper, and I work on and Joe, who will talk next, will present that work, some of that work on Afrotemperate, a group of Afrotemperate daisies. And when you just map their distribution, it looks very similar to a Rand Flora distribution. But these are plants that have a cool growing season. They grow at very high elevations, except down here in the Cape, where they get down to sea level. And so, I just thought it was really interesting that you pulled out the ecology, and then you had that separation of the arid adapted and the tropical adapted taxa.
Right. So we're also aware of this, of studies of Afrotemperate taxa, like you say, the daisies, and there are several groups showing that some of those groups, they originated in Southern Africa, and then they moved to the northeastward, and they use the Eastern African mountains as a stepping stone or a bridge. those groups, we actually don't think we didn't consider them Rand Flora. At the beginning, we have lots of candidates for the Rand Flora because there are even groups that are more arid adapted, for example, in Segophylaceae. And it was difficult to decide even the Dracaena Draco, the famous Dracaena trees and everything. So it was difficult to decide what is Rand Flora. We have a lot of discussion with Peter Linder on that and with other South African biogeographers. For example, about the dry diagonal, those groups showing dry diagonal pattern, are they Rand Flora? So at the end, we decided that we constrain Rand Flora to groups that are more xeric adapted. They are also subtropical, but those Afro-temperate groups that show that pattern of relationship between Southern Africa and Eastern Africa and the North, and they seem to show the pattern of northeastward migration. We didn't consider them Rand Flora, neither those that are, for example, Erica, neither those that are dry diagonal, so xeric adapted. But I think it's a question of the idea of extinction. In Afro-temperate groups, I don't think that extinction has played, you can correct me, that big role. In the Rand Flora groups, it really looks like they are very old. They have all these junctions. There have been ecological vicariance. We will reconstruct the ecological, the niche tolerance, and also that they have a pattern of divergence that seemed to fit climatic events that were linked to acidification. But I agree that it's sometimes difficult to define what is Rand Flora, actually.
No, well, I think I agree with you completely. I think they are completely different floras that just superficially look similar.
Literally a juxtaposition of course, Africa is the flora of Africa is very complex and it has a lot of events. You cannot simplify in one thing. Even the Rand Flora is not one event. It's not what they thought at first. Huge macroflora that was split by aridification. Now we know it's not like that. But still, it's very interesting in the sense that you can study extinction at clade level and across many different groups.
Isabel,
Hi.
my name is Conrad. Thank you very much for a very nice talk. I really enjoyed it very much, especially because I'm not a botanist, I'm a zoologist. And I would just like to point out something that I find extremely interesting in your talk is I worked on a tick, one of my MSc students a while ago, and it's a very generalist species. And this southeast West African clades is exactly the same in this tick that we then account to vicariance, and the link between East and West Africa also seems to be much stronger than between East and South Africa. And the clades fits perfectly. And this is a generalist tick occurring on at least 20 different antelope hosts and warthogs. And those hosts also show similar patterns. So there's very strong evidence for vicariance on the animal side that is congruent with what you actually find in your plants. And I just thought, I wish I can do all those fancy models you're doing because it will really help me to actually answer quite a lot extra, but very nice, thank you. I just wanted to actually mention that.
Thank you, that's quite interesting because what I heard from fellow zoologists, they mentioned the northwest, northeast pattern in Africa. So they have the Sahara barrier, but I haven't heard about the southern connection, southeast and west. So that's very interesting. And I wonder, I mean, I'm sure there are patterns that are very similar. I just wonder if they have similar explanations or driving processes. I mean, if extinction has played a role on that, what do you think? And you mentioned the hosts. You think that maybe, I mean, you know, for example, how old is your pattern? Where the divergences?
It's approximately 4 to 7 million years. And we coincided that with actually rifting in East Africa, which had a peak in approximately 5 million years ago. And that East Southern African process is very well known. It's well known for spring air. It's present in many antelope, it's present in carnivores, it's present in chameleons. So you find this East Southern African break being very strong. And we always blame the Brachystegia woodland and then also the rifting. And then in terms of the ticks, it was also tropical and more temperate. And that will then also give different breeding cycles in these ticks. And because they breed at different times, they probably can't hybridize even if there is contact. So if there's host movement, they'll be out of sync in their life cycles. So we just thinking, we don't know. I mean, it's one taxon, it's one species that we looked at, but the hosts are really cool because they show similar patterns.
Yeah, I mean, that's very interesting. You mentioned that in hybridization and that they are out of, I mean, they seem to be in a way evolutionary isolated. And the dates you mentioned of 7 million years, 4 million years, 5 million years, they fit very well our dates.
Exactly.
Many of the Rand Flora groups, for example, Cantoloma rotundifolium in Namibia, and Cantoloma canariense and Cantoloma riveriflorum in North and Northeast Africa, they have this 5 million year divergence between the south and the east-west, and then 3 million years. And some Flocama, for example, is 7 million years, and Cleinia is 7 million years. So they seem to fit very well the idea that they are Miocene-Pliocene groups. This is not the Pleistocene. They are not reflecting at all Pleistocene events. Some of them they do, but in general they are pre-Pleistocene. So they are, and they seem to be linked to the idea that they use the wetter periods in Africa to move around. It's not that it's pure vicariance, there has been some dispersal. And they also have divergence close.
Thank you very much.
Thanks. I need to go and study the animal examples.
Michelle has got a question.
Yes, Michelle.
Thanks, Isabel. That was a very interesting presentation. I was very interested. I think you only really showed one of your SDM maps, but I was very interested to see that your modeled predictions were actually not very good fits for your actual presence data. And I was quite interested to see that because I've done some SDMs for acacias, and I've seen SDMs for other African taxa. And generally, climate isn't a very good predictor for the distribution of many African taxa. And of course, this could be partially because of the fact that you've had these expansion and contraction events. But it could also be because I don't know if you've included fire dynamics or anything like that in your model. So I was just interested to hear if you had thought about that and what your thoughts were, and whether this was a general pattern that you found that your SDMs were not very good predictors, or whether it was just the one map that you happened to show. Maybe it's a bit of an unfair question.
Right. I think there was the map where I show all those different niche models in a,
So it's the first niche model, the first right at the start of your presentation where you showed one of your niche models, and you said that's one way to predict.
Right. Yeah. That was
the species I think was sort of on the mountains, but it was also predicted to much of the coastal area of East Africa.
Yes, I can say that our SDMs are not very accurate in the sense that there is one thing we are, if we build SDMs based on present day data, so with present day climatic data, then they come sort of well, but because we also plot backwards in time into the Miocene, 50 million years, and Pliocene, then our climatic data, which we got from a climatologist in Sheffield, they have very large ranges, size 100 kilometers, so the resolution is very poor, doesn't capture at all mountains or islands, so the patterns are fuzzy. And of course, because we plot backwards in time, and we have very few ancestors because of extinction, the resolution, as you mentioned, is quite, is not as accurate as, for example, you get when using, I don't know, present day data or Maxent. We mostly use ensemble forecast, as you probably do, and here we have a few tweaks to correct for rare and endemics, where we have very little data, and it was extremely biased for certain areas. But it's true, they were not the most accurate SDMs, but it was enough to see that at least that there was that connection between East and West, across North Africa, and sometimes between Southern and Eastern Africa, across Central Africa, and the position and the time of those connections, it fit quite well, the climatology of Africa. So this Miocene desert and then the tropicalization, but you need to take it, with, 50 million years, 20 million years ago, and with ancestral species, right?
Yeah, thanks. I was just interested because it's a pattern I think we find across Africa, and I think it's quite different to a lot of other continents where climate is actually a very good predictor of the distribution, but very interesting. Very interesting.
Yes, that's true. I missed that part of your question. That's totally right. We are using, for example, for present day, we do use altitude, we do use solar radiation, we do use soil, but for ancestral SDMs, we only have temperature and precipitation. That's all we have. So that's true. We miss a lot, and fire is an important component that has a lot of a big role. So that's also one factor that we are clearly missing in our predictions.
Very nice. Thank you very much.
Okay, and I've got one more question for Isabel. You mentioned the use of, I think it was neural networks in looking at SNPs, and I wondered if you could just briefly explain a bit more about that and how that works.
Yeah. I don't have my slide anymore, but maybe I can show. Or shall I try to share? Yeah.
Yes.
Yeah. Okay. So it's next slide before. Yes, this one. Yeah. So what we do is that we use usually when you have SNP data, you use simulation-based methods. For example, approximate Vagisian computation, ABC methods, and you try to estimate statistics from those SNPs. For example, heterozygosity or nucleotide diversity or FST or different statistics for the populations. But then you miss part of the data because it's only one statistics or maybe a bunch of statistics. So we wanted to use the entire structure of the data set. So SNPs, you have this matrix of SNPs and you have them coded as 0 and 1, the minor and the major allele, and you want to capture the image of those zeros and ones. It's like you have a picture of zeros and ones, like a binary code, and you want to capture the structure. How do the zeros and ones distribute? Which are the sites, segregating sites in the sequence? And with convoluted neural networks or machine learning, you can actually do that. You can take the entire image, which is the covariance and variance between SNPs in the sequence and across populations, and you translate that into information. And how do you do that? So these convoluted neural networks, you have a series of layers in the network, like algorithms that decompose the information into different ways. And you have, like in the slide on the left, you have the information go through the different network layers, and they are trying to reduce that complexity into dependencies between the segregating sites. And so it's similar, for example, those working with SDMs, working with Maxent. Maxent is a machine learning. So you actually never see the actual livelihood of the dependencies between sites. You don't see a formula, but it's something in the black box, and the machine learning is able to capture, decompose the image into information. And at the end, what you want is that you will be able to do that for different scenarios. So you have your SNP matrix, and then you have a SNP matrix you simulate under a scenario of bottleneck, under a scenario of geographic expansion, like in ABC. And you do the same decomposition in the neural network, and then you compare the observed against the simulated. But you don't do it based on one statistic, one value. You do it based on that image or picture or variance, covariance between segregating sites in the SNP matrix. So they say that machine learning in that sense is more powerful, and it has been shown that it's more powerful for model choice, to choose the right scenario. like, but not maybe that much for parameter estimation. Like what is the rate of mutation? What is the rate of migration? That's a bit more difficult. So now what we do in this slide here, I show we combine ABC and convoluted neural networks. And I must say, I'm still fond of inference methods, Bayesian, and so I like to see the actual livelihood formulation. I got to see the mathematics, and here I don't see it. But I agree that this is very powerful to introduce complexity that we cannot do right now in model-based phylogeography.
Yeah, wow. That's a great explanation. Thanks, Isabel.
Thanks, Nicole. It's really difficult to say that in one slide, but
absolutely, yeah. Well, that's fascinating. Okay, well, maybe we should, unless anyone's got a pressing question, I don't see any hands up. So I think I'm going to close the question time then. Just say thanks very, very much, Isabel, for a great talk.
thank you.
And now we'll go on to our second speaker.
Yeah, I mean, I'm just sorry, I'm just frowning about something else.
Sorry, is that a mic that's off, Lisa? Yeah, I think so.
I need to turn off my mic. No, yes.
No, that wasn't you. I think it, yeah. Okay.
People
So,
people should just watch out. The mics come on. We constantly muting people as these things happen. So just watch out for your mic and make sure that it's off all the time.
yeah, thanks, Farnes. I should have actually announced that at the beginning. And also just to mention to people that they're also welcome to type questions into the chat box during the course of the presentations, and we'll check up on them afterwards. Okay, so I'm going to introduce Joanne Bentley now. She is here, but her talk has been pre-recorded, so Tuan will play it in a moment. Joanne is a postdoc at the African Climate Development Initiative at the University of Cape Town. So she's going to present work that's kind of from her previous life, where she was actually doing a master's with myself and Tony Verboom. And this is a biogeographic investigation into this Afrotemperate daisy lineage. Okay, so over to Jo's presentation.
Hi everyone, thank you for coming to listen to my talk. My name is Joanne Bentley and I'm presently a postdoc at the African Climate Development Initiative at UCT. So the work that I'm going to present to you today is actually from my MSc, which I did with Nicola Berg and Tony Verboom. The title of my talk is Lineage Origin, Historical Migration Patterns and the Contributions of Geographic Proximity and Ecological Filtering to Dispersal in an African-centered paper daisy lineage. This talk is about a group of about 120 species of very morphologically diverse paper daisies, or everlastings, shown here in the bottom right of the slide. These daisies belong to the Nepheli tribe and are believed to be the earliest diverging lineage of Nepheli. This earliest diverging lineage contains several genera that, based on our earlier molecular phylogenetic analysis, now represent the genus Udera, and so I will be referring to this clade as the Udera clade for the rest of this talk. This clade is particularly interesting from a biogeographic perspective, with members distributed from the greater Cape Floristic region in the south, in the more arid Namib and Karoo regions, throughout the more mesic and high elevation Drakensberg grasslands, and along the East African highlands, as well as in the greater Mediterranean region in the north. If you have a look at the distribution of the group, large geographic range disjunctions are very apparent between closely related species. To explain these disjunctions, it has been suggested that historically, closely related lineages might have migrated along specific routes within their environmental niche, with the Mediterranean and more arid lineages in the greater Cape Floristic region, the Namib and the Mediterranean, migrating along transient corridors of arid habitat that are believed to have existed periodically since the Miocene and that coincided with periods of aridification. Similarly, Afrotemperate lineages in the Drakensberg and East African highlands might have migrated along stepping stone mountain peaks of suitable habitat, which coincided with the creation of these high-altitude habitats during times of tectonic activity during the Miocene, and particularly during the Pliocene. Southern Africa has been identified as a potentially important source of floristic diversity globally and of arid land biodiversity in Africa. In addition, it has been identified as a likely center of origin of asteroideae. By generating a time-calibrated phylogenetic hypothesis for this clade, we aim to explore the center of origin of the Uruguay, the directionality and timing of its range expansion, as well as the relative importance of geographical proximity and environmental similarity as determinants of its dispersal. We predicted a southern origin for the Uruguay, with northwards dispersal to the semi-arid Mediterranean, potentially during phases of heightened aridity, and to the highlands of East Africa during phases of continental uplift. Given that the Uruguay comprises elements that associate with arid environments on the one hand, and high elevation mesic environments on the other hand, we predicted that ecology might influence its dispersal significantly, resulting in higher rates of dispersal between environmentally matched areas. Here is our dated phylogeny and biogeographic analysis. What we found was that the ancestor of the clade originated in a relatively mesic habitat in the south, and then subsequently diversified into surrounding arid and afrotemperate habitats, as and when these habitats became available, with the clade accumulating most of its diversity in the last 10 million years. This supports a southern origin for the clade, as well as several dispersals along supposed arid tracts, from the Cape to the Namib and then to the Mediterranean, and on afrotemperate tracts from the Drakensberg along East Africa, with some examples of back colonizations from the Mediterranean into the East African highlands. These dispersal events were found to be synchronous with estimates of heightened aridity or tectonic uplift in the area, and also match the timing of predicted arid and afrotemperate tract migrations in other lineages. Historically, it seems apparent that these lineages tend to track similar habitats. For instance, migration occurs from the Cape to the Drakensberg to the East African highlands along high elevation habitats, while the arid lineages move from the Cape to the Namib and Karoo and then to the Mediterranean. We hypothesize that geographic proximity is likely a significant predictor of range evolution in the Uruguay clade, as migration appears to be more likely to occur between areas of contiguous habitat than between great distances. There also appeared to be a tendency for niche conservatism, in other words, for species to retain ancestral niche characteristics. To test our hypothesis, we georeferenced specimens to calculate pairwise geographic distances between the species in each of the 5 major centers of diversity, and then also calculated pairwise climatic distances between the species based on 19 bioclimatic variables. We did this to derive metrics that could be used to quantify the contributions of geography and climatic niche in dictating species ranges. We then used GLMs to assess the significance of climatic and geographic distance as predictors of between-region dispersal rate. The results of the GLM strongly supported geographic proximity as being an important indicator, while to our surprise, climatic distance was not. However, our impression was that members of this clade are still selecting to disperse within a particular environmental niche, and that possibly the bioclimatic variables that are dictating the niche don't differ substantially across the clade's distribution. To explore this, we also sampled the bioclimatic variables from a randomly distributed set of species points from within a convex hull polygon enclosing the entire species range, and compared the extracted bioclimatic variables with those from the actual species points. We then did a principal components analysis and compared the scores of the species points and the random points on the first 2 axes, which we found to differ significantly, showing us that there were specific bioclimatic variables associated with the species distribution. We then had a look at the eigenvalue scores of these principal components and found that Uruguay clade members disperse within a moderate temperature and rainfall range, avoiding the more extreme climates. Seasonally and diurnally variable environments are also preferred, represented by bioclimatic variables of isothermality and precipitation and temperature seasonality. Overall, there is a strong signal that this clade sticks within a temperate climatic niche. So, to wrap up, our data infer a southern origin for the Udera clade, with multiple dispersals along arid and afrotemperate corridors. We show that these species disperse within a particular ecological niche, and that this niche is a temperate habitat, with the more extreme environments being avoided. We also see this in the historical signature, with species extending their range only as suitable temperate habitats became available. However, within this particular climatic envelope, rates of dispersal between regions are very much influenced by geographic proximity.
All right, thank you very much, Joe, for a very compact and information-packed talk. I see, Joe, you're available in, yes, you're here to answer questions. Okay, so I'll throw it open to the floor again, see if there's any questions or comments from the audience. And Joe, maybe are you prepared to put on your video?
Yeah, and I have one question. Yeah.
Sorry, I just need to move. Hold on.
John?
Yes, go ahead, Isabel.
Okay, I see. I thought that was very interesting and much in tune with what I showed, the idea of the geographic proximity versus climatic distance. You at the end, you sort of conclude that this clade, they use both the arid tracts and the afrotemperate dispersal route. So do they have, I mean, the arid tract is very different to the afrotemperate dispersal route. So do they show different climatic affinities within this clade? It's like you can find, I didn't get that part, you can find different subclades. One is more arid and the other more afrotemperate, or they are both using this eastern mountains dispersal route? Or?
Sorry, I can't put on my, well, I'm not sure that I should put on my video because my internet connection is quite stable. So I'll just talk if that's fine.
Yeah, that's fine.
So there are definitely members of the Udera clade that are more arid lineages, and those are not, they're distinctly not afrotemperate lineages. So there is a distinction within the clade between those species. I don't know, Nikki, if you have something to contribute to that.
Just that I think if I remember correctly, the arid adapted ones are all annuals.
Right. And Rosinia?
The Rosinias are all perennials.
Okay.
Yeah.
So when you mentioned that they all have this temperate niche, so they avoid extremes. You refer only to the afrotemperate niches or also to the arid ones, the arid tract? Because I always thought the arid tract was more dry and extreme, and so like this Sagophylaceae and Rosymbium, or is it like they are both very niche conserves? They are both very niche conservatism, and they maintain this avoiding the extremes, or?
I guess while there's definitely a more species that belong to more arid components, I guess they are still selecting to disperse within an environment that isn't too extreme. We're not talking about desert adapted plants here. We're talking about semi-arid plants that occur in the interior of South Africa and in the Namib, but yeah, still avoiding the more extreme environments.
Right, that's very interesting. And when you mentioned the this geographic proximity versus climatic distance, have you tried, Nicole, Joanne, about the measuring the rate of climatic variance in the phylogeny versus the rate of dispersal, or the rate of how far do they go per unit of time versus how different the adaptation, did they change actually, even though they are niche conserved, do they show variance, and then you can plot that on the phylogeny, so comparing the rate of climatic variance versus the rate of dispersal or the rate of movement.
Yeah, that isn't something that we've done. But yeah, that could be, I mean, this is still, yeah, suggestions are very welcome, because yeah, this is actually, even though it was done so long ago, it's actually still a work in progress. So yeah.
It's a group quite interesting. Yeah, fascinating.
Thanks.
Yeah, are there any other questions for Joe, or comments? Yeah, I think that would be an interesting analysis to look at. I think one of the things that's held Joe up a bit there is redating everything using a different method. So we won't redate again, but the dates are there for testing those kinds of hypotheses.
Yeah, that could be, and the idea of usually we have this distinction between ecological vicariance versus geographical vicariance. In this case, is dispersal. So do they become allopatric, but they still preserve the niche? So they have the same niche as they move on in dispersal, and sometimes you see that maybe at the extreme of the dispersal track, sort of, they have a different niche, and then you wonder, did they adapt in situ to that climatic condition, or did they diverge earlier? So that's also very interesting. We have one study in which we analyze the idea of geographical speciation versus ecological speciation, but there is also this ecogeographical speciation in which they become separated because of dispersal. There is a distance between populations, no gene flow, and then later on they adapt to different conditions. So it looks like they are actually climatically separated, but the adaptation to different climate was in situ. It was not the driving force of the speciation.
Yeah, so we did actually do another analysis on one of the groups within the Udericlade called Macroevolutionary, which is a Drakensberg and East African Highlands endemic group. And yeah, in that analysis, we did look at ecological versus non-ecological speciation, and tied that in with geological, with tectonic uplift in the region and morphological similarity. Yeah, so kind of along the lines of what you're describing now. Yeah, so we have done that in a component of the Udericlade, but yeah, I think it would be nice to look at something like that clade wide as well.
Yeah, I would be very interested to read that. Yeah, I will look for it on.
Sure.
Okay, thank you very much, Joe. And now I think we'll move on to our next speaker, it's also an Afro-temperate group. So our next speaker is Seth Musker, who's doing his PhD currently. He's based in Germany, and he's a South African who's doing a PhD in Germany and on this African group, and he's co-supervised here at University of Cape Town. And I think his title was discovered during a journal club. Am I right, Seth? Over to you.
Absolutely, yeah. Thanks, Nikki. How's my sound quality? Can you hear me fine? Okay, good. Let's get this started. How's that?
It hasn't come through yet, but maybe there's just a lag. It's not sharing. We can see the tree printed on your t-shirt.
Right. Okay, that's not right then. This worked earlier, perfectly fine.
Or if anyone has any other questions for Isabel or Joe, we could just quickly slot them in while Seth. So I don't know, Seth, I don't know if we can help you from our side.
Yeah, this is very strange. One minute.
That's looking good.
Yes,
Yeah.
There we have it.
It's like it's witchcraft. Anyway, thanks. Ready. So yeah, first of all, thanks for coming, everyone. Yeah, I'd just like to mention and thank my supervisors, Nikolai Nurk, Mike Perry, and Tony Verboom, as well as the funders, the DFG. And also just mention that this is ongoing work, so I very much welcome feedback and ideas. So how should we study speciation? I think Isabel has already given us a very nice look at this continuum between micro and macroevolution. I think Sarah Villa, in this 2009 article, provided a nice simple perspective, which is that we can basically choose to study systems in which the speciation process is complete, or systems in which it is incomplete. The former has limitations because the factors that cause speciation can become obscured by evolutionary change over time. And if we choose to study the latter, then we need to find an appropriate system where multiple factors could be at play. So in other words, we have to seek out inherently difficult problems where we can't be sure what to call the things that we're calling things. So that brings me to Erica. It gives Ericaceae its name. About 800 species worldwide. They originated seemingly in Europe and dispersed through the Afro-temperate regions, eventually making it to the Cape roughly 10 million years ago, at which point they experienced an explosive diversification. You can see from these red and yellow colors, indicating diversification rate estimates. This is a study. This is adapted from 2 studies by Perry et al. And so we have almost 700 species in the Cape, making up a large proportion of the genus. One of the very notable things about this Cape clade is its great phenotypic diversity. In particular, we have seemingly repeated convergent evolution of diverse pollination syndromes, including bird, generalist insect, long-tongued fly, for example here, Erica jasminiflora, wind dispersal, these short tube things, and even some rodent dispersal, although that's quite rare. And that's just pointing to the Cape, so you know where we are. And so for my project, I've decided, I've decided to focus on a somewhat smaller problem and look at the so-called Erica abietina viscaria clade. It doesn't have a better name, so it's just bookended by the first and last in the alphabet. It's taxonomically complex in that there are 19 described species currently, but 30 taxa, including subspecies, as well as several varieties, which could also represent distinct species. And it's just like the genus as a whole, floristically diverse. including both within and between species. We see these various color gradients from white, pink to red, sometimes all three, yellow to orange, and even some green flowers. We also have differences in flower shape. So we have these tubular flowers, long tubes pollinated typically by sunbirds. This orange-breasted sunbird is a Cape endemic and a very important pollinator in general, especially for Erica. And then several generalist insect pollinators, probably bee-pollinated species. And this is just a geographic context. It's widely distributed in the Cape Floristic region, but it's most diverse in the southwest, which is a very common pattern in Erica and in the Cape Flora in general. Many of the subspecies are parapatric, sometimes even sympatric. There's some evidence for elevational stratification between closely related species, and there's also some evidence for edaphic partitioning between closely related species. Traditional phylogenetics, as expected, failed to produce a well-resolved phylogeny. However, Piri et al. 2017 were able to show that several subspecies were indeed distinct species.
Oh, yes, Seth.
Yes, I am. So, sorry about that. Basically, so in particular, they were able to show that Erica abietina was paraphyletic. And so they were able to resurrect Erica grandiflora, and they described two subspecies, and they described a new species, Erica situs himalis, from the Grootwinterhoek mountain range, which had been described as a subspecies of Erica abietina, but is indeed quite distinct. And so there are 4 subspecies which remain to be conquered, and those are this sort of comb-like structure over here, what I'll refer to from now on as Erica abietina, along with a couple of outgroups, both of which are also endemic to the Cape Peninsula. So on the subject of the Cape Peninsula, it's a mountainous so-called continental archipelago, and it's isolated from the rest of the Cape Flora, the Cape Floristic region, by a vast tract of not just Cape Town, but also mostly sands of alluvial origin, which are inhospitable to most Ericas. And so we have these 4 remaining subspecies: Erica abietina, the nominate, with red flowers over here. It's confined to Table Mountain, and it also has these long tubes, so it's sunbird pollinated. We have Erica abietina atra rosea, with pink flowers and long tubes. That's down here. That's mostly in the south, but it also extends in this arm of the eastern slopes of Table Mountain. And then we have Erica, the two short-tubed subspecies, Erica abietina constantiana, with pink flowers, short tubes, and a distinct, beautiful, really lemony scent. It's mostly found in the central area. And then we have Diabolus, named after Devil's Peak, with a medium-length tube, also pink. There's no scent. The pollinator is uncertain, but it's probably also bees. And it's very range restricted, all the way up in the north. So the approach that I took used genotyping by sequencing, which is a genomic sampling method. This is just an overview of the sample sizes for the various sort of a priori groups. And we aligned the raw reads to the Erica ceranthoides draft genome, which came in handy later. I did various analyses. I'm just going to present a few here which tell the sort of story that I want to tell. The first is principal component analysis, which tries to find basically as much signal in the data as possible out of all the various noise. An admixture analysis, so model-based, explicitly attempting to model the process of allele sharing and gene flow, for example. And then admixture graphs, which is sort of can be thought of as a phylogenetic approach. And then at the very last, I'm going to talk about this selection analysis, for which we also use PC angst. So here's a little bit of geographic context again, and I'll refer back to these maps, but basically this map on the left is a simple stamen map, but it shows quite nicely the sort of topographic heterogeneity that we see. And then these are my sampling sites. So again, we have the same key. You also see the geological variation here. So the three main substrates on the Cape are shale up in the north. The granitic soils, also low elevation, and then these Table Mountain sandstone soils, very nutrient poor and very, very distinct. And then here we see the elevational heterogeneity as well, increasing towards the north. So on the PCA results, we have the same color scheme again as the maps. And overall, we see a decent amount of signal. So we have these sort of groups with somewhat with disjunctions, and that's always a nice thing to see rather than a single blobby mess. But there's a lot going on. So let's break it down. First of all, on the sort of lower end of the first principal component, which explains the most variance, about 8%, we have abietina plus diabolus, and they're characterized by having these hairy sepals, the leaves quite densely packed, and having this northerly distribution on sandstones. And they're distinguished for obvious reasons in pretty much in only the floristic traits and nothing else. Then we also see this interesting unexpected result here with subspecies Atrorosea showing two distinct genetic groups distributed in the north, which are these in the center of the principal component space, with these tapering to a point sepals, narrow leaves, you can see here, and they seem to be confined to wet shale and granite slopes on the east slopes of Table Mountain, and somewhat further, a little bit further south. And then we have the southern widespread population. Unfortunately, I have quite sparse sampling, but they are also quite morphologically distinct. They have these much more robust leaves confined to sandstones, and they have these less tapering, more obtuse sepal tips. And then the mess. So there's evidence of hybridization, and so these yellow dots basically are indicating the fact that I actually thought these might be hybrids, and that was based on floral traits primarily. So, for example, here we have collection 478. That's this one here, which I classified as Atrorosea based on its long floral tubes without any bulge. Then we have these typical Constantiana in this region with short tubes and lemony-scent, and these hybrids with sort of intermediate features. And it doesn't seem to be a geographic thing. Many of these are occurring right next to each other. So it's possible that this is a secondary contact zone forming a hybrid swarm. A much clearer example of hybridization are these two specimens from the north, which appear to be hybrids between Erica abietina, subspecies abietina and subspecies Atrorosea, the northern population specifically. Admixture analysis produced similar results. The best K was 5, so K being the number of genetic clusters. Atrorosea north is recovered nicely with these green bars. The hybrids are detected that I just mentioned. However, it cannot distinguish between abietina and diabolus, but a look at the model residuals suggests that in this case it's a poor fit. And in particular, these negative residuals indicate that there should be a split where there is a lump. So these should, based on the residuals, they seem to suggest that these should really be distinct in some way. But this particular model fails to uncover that variation. And then there is this sort of mess again between Atrorosea and Constantiana, probably due to the secondary contact effect. And there's also a possibility for an isolation by distance effect, which I won't explore now. Moving on to the admixture graphs. So we can see that we're using this Erica Nevillii as the closest outgroup, very well supported as the closest outgroup relative. And there are a couple of things to note. So I removed any individuals showing signs of recent admixture. So any result here should reflect ancient admixture, ancient gene flow. And a resampling approach suggests that this is the best tree. It predicts the data a lot better, significantly better than a tree without any of these reticulations. And so in terms of the tree itself, the graph itself, note the lack of a sister relationship recovered between Atrorosea south and north, as well as the obvious contribution, the reticulate sort of origin of this northern population, with a small contribution coming from abietina, the long-tubed form, and most of the contribution coming from the most recent common ancestor of this northern Atrorosea population, and Erica abietina constantiana. So the selection analyses, basically what we did was we focused on abietina and diabolus, because they're very closely related, but they have different phenotypes. So we have red versus pink flowers and long versus short tubes. And using BLAST on the loci to which these highly differentiated loci aligned in the Erica ceranthoides genome, we were able to uncover two decent hits, and that are particularly interesting. The one hit was to a flavanol synthase gene, and flavanol is a very important compound involved in generating pigments. Flavanol itself is a pigment, so and usually involved in the formation of red coloration. So differentiation in this gene could explain this particular divergence. And then this other interesting one, which was actually pretty exciting, is these microtubule associated protein hit. And I'd never heard of microtubules, but apparently they are cellular structures involved in elongation and during the growth of the cell. And so unfortunately, I don't have good microscope images here, but I have observed that in the long-tubed species, the cells themselves are elongated, and that seems to be what's causing that elongation of the tube. And it could be that this is a very important gene in general in Erica in determining these pollination shifts. So in conclusion, as expected, Erica abietina is a very complex system. There's cryptic diversity, which is always exciting. There are at least 2 independent pollinator shifts from between sunbird and insect. The direction is unclear. Both geographic and ecological forces probably drove and continue to drive divergence, genetic divergence. Geographical contact zones are very important in predicting the occurrence of hybridization. But there's also evidence of ancient admixture, which may have driven adaptation and divergence, in particular in this northern Atrorosea population. We then also hypothesized that abietina and diabolus in the north are in the very early stages of speciation. And finally, in conclusion, I still don't know what to call these things. So thank you very much, and feel free to contact me again for any, if you feel so inclined. Thank you.
Thank you very much, Seth. That is quite an amazing system. Thanks for a very clear talk.
Yeah.
Unfortunately, no one gets claps after virtual talk. It's one of the things we miss. We can use our little reactions, I think.
Yeah. Sorry, I realized I have to turn my camera back on. So
that's great. No, thank you very, very much. Okay, I'm going to open the floor up again. I'll look for hands. Not clapping hands, but hands up for questions or comments on Seth's talk. And just while you think about it, I just want to comment myself about how absolutely astounding it is that this variation is on a tiny piece of land. I mean, the peninsula is what, how many kilometers from top to bottom? Is it
I think it's about 40 kilometers, yeah.
40 kilometers.
They should have put some scale bars in there.
Within a major, within a major urban zone as well, which is quite frightening if Erica is sort of representative of other Feindhorst groups.
Yeah, indeed.
And also that in this hyperdiverse flora, there's indications that possibly it's understudied taxonomically. But yeah, I guess my question there is the original taxonomy, and then I see Kelsey's got a question. So were they kept as subspecies rather than species because of intermediates, or what was the situation?
They weren't actually kept as subspecies. It was complicated. The abolus seems not to have been described at any point. So it was given a new name, but basically it was all a single. It was, so we had Erica abietina, we had Erica felicifolia, which was Atrorosea, and then Erica conica, which was Constantiana, and Oliver in 2001 lumped all of those into Erica abietina, as well as described subspecies Diabolus, and at the same time they lumped grandiflora and Portfoliosa, and they described this new, well, what has turned out to be a new species. They described it also as a subspecies of abietina. So they really expanded abietina a lot. And my job is to kind of continue the work of Mike Pirrie to some extent in breaking that back down again, slowly chipping away at it. Yeah.
Okay, I see there's a question from Kelsey here, and she's put it in the chat. She says she's not going to use her microphone due to competition in noise in the house. But she's wondering about your flavone gene. Did you test for selection? She says she might have missed it, the flavonol synthase gene.
Yeah, so it's a principal component-based approach, and basically it aims to find SNPs which contribute significantly and have a large effect size in differentiating groups along the principal component axes. So I didn't actually show the plot, but basically we have the first principal component, which explains most of the differences between Diabolus and abietina, and then we look at SNPs, the SNPs that basically contribute the most to that axis. And then in identifying the flavonol genes, it involves basically pulling out the chromosome or the contig to which those loci originated from or were aligned to in the 730s draft genome. It's a very draft genome, which I put together. But it, and then also finding loci that had not just a single SNP, but showing this pattern, but several SNPs. For example, and also those with the strongest effect. So yeah, thanks.
Okay, Kelsey writes, I see, very cool.
Yeah, nice.
Okay, yes, Isabel.
Yes, I couldn't find the button for raising my hand, so I did it actually physically. That was a very nice talk. It was a fascinating study system because you show both morphological disparity and ecological divergence and geographical divergence. So at the end, what I got is you seem to conclude that it was both geographic and ecological, or ecological adaptation and geographic isolation. But then you mentioned these morphological differences in flower shape and color, and they seem to be driven by this pollination system, the sunbirds. So the thing is, what came first? So how you know this is a case of, for example, neutral divergence driven by geographical isolation or even ecological adaptation to different environments in different geographical regions, or was it reproductive speciation driven by this pollinators, the birds? For example, do these birds live in different regions? There are birds here and birds there, so there are different species pollinating, and do they?
Yeah, so I mean, I think that pollination shifts are very important, but may be contingent on other changes. So I think that most likely there's the history. So what I'd like to look at further is some sort of demographic modeling, because I suspect that, for example, in Abietina and Diabolus, that there was a population bottleneck in Diabolus associated with some perhaps climatic changes, as well as the fact that it's this sort of edge population. So perhaps it got isolated at some point, and that some founder effects are what essentially allowed it to shift into this this different pollination niche. And so yeah, and that's work that's still ongoing. But yeah, there is no single answer to those questions. And unfortunately, I think that, you know, despite the fact that we have genomic data and it's pretty rich, I would love to have more to answer those sorts of questions, especially more contiguous geographic sampling. It's unfortunate that Erica is very difficult to get good DNA out of, and that's actually what limited me to a large extent in generating this data. Yeah,
we have some experience also with difficult DNA in some Rand Flora taxa.
Yeah.
But I was thinking on when you mentioned selection, those genes that you found that they have a signal of positive selection, and they were linked to the, or you thought that they are linked to the difference in morphology. Have you looked for, I mean, were they the only genes that you found signal of selection? Because we use target sequencing in our genomic approach. So we actually identify the genes, and we have genes that are more involved in climatic adaptation. So they are more involved in conferring resistance to dry habit, and other genes that are maybe more involved into the morphological divergence. Or so I wonder if you will find also genes involved in the ecological adaptation, or so that will tell you that there are different answers, yeah, or a mix of answers.
Yeah. I mean, it's tricky with a draft genome and this sort of blast approach that is kind of a shot in the dark of sorts. But on the subject of how of how many of these loci we had, basically what I did was I found the sort of top 1% of loci in terms of their magnitude of effect, as well as contigs or chromosomes with several of these SNPs. So there were only a few, actually, there were only 5 that had more than 4 SNPs in this category, in this top 1%. And so among those 5, 3 of them came up with nothing, and 2 of them came up with these 2 genes that I presented. So I'm fairly confident that these genes are under selection. But of course, it's hard to say for sure. And the beautiful thing about target capture, we're actually, the next one of the other things I'm doing for my PhD is target capture, and we've added these loci to the custom bait set. So we'll be able to get those loci hopefully in full for not just abietina, but for the abietina viscaria clade as a whole, and perhaps yeah, I'm aiming to do some sort of selection analyses, looking at transition-transversion ratios, for example, across the clade.
Yes, that's what we do. Yeah,
yeah, it'd be really cool. Waiting on the data still.
Yeah, I mean, Erica is a fascinating study system.
Thanks. I made it myself.
The other thing I noticed, you're talking about selection, Seth, the You had some soil differences. And
Yeah,
of course, I mean, apart from, there's so many things associated with different soils, but there's also very strong pH differences, which are probably quite strong even on the peninsula. And I mean, I don't know if there's specific adaptations that plants would have to make.
yeah. I wish we had more detailed soil maps. I know we have much more detailed soil maps than we did 2 years ago, but for such a fine scale as the Cape Peninsula, it's really tricky. I guess so. In terms of pH, do you mean that there's a strong that pH is decoupled from underlying geology?
No,
Or would
I
that
mean
be
linked to the
in the soil?
underlying
Okay.
geology, like the
Yeah.
very acidic quartzites and the probably, I think, more neutral granites and shales.
Yeah, yeah. Certainly, it's a good proxy for nutrient levels, and they probably do present very different selective environments. But I think there are the sample size isn't really large enough to disentangle the effect of, for example, pH versus climate versus mineralogical properties, etc. So at the moment, those sort of observations are somewhat anecdotal, unfortunately. But I do think that they're cool, so I'm sticking with them.
Yeah. Okay. Well, you know your system. You get to know the system. So. Yeah,
I mean, after a couple of years, you become familiar with these things. And it was, I had an idea beforehand that these two Atrorosea populations were distinct. And so it was kind of quite cool to actually see that in the genetic data.
Okay.
Any more questions in the question box, in the chat box? So I don't know if there's anybody that still want to raise their hands. And just unmute and ask the question.
I'm hearing something, but I don't know if it's very garbled. I don't know if it's just an accidental microphone. Yeah. Well, I think maybe we'll just open things up for a general discussion. If anyone wants to talk about, make any comments or observations, or have questions for any of the three speakers, we can just chat about any questions that might come up or topics. Seth.
Yeah, so actually, Isabel, I was wondering about the Rand Flora. So I think it was something that Conrad said about seeing the same pattern in the ticks and the hosts, and I wondered if something similar might be present in the Rand Flora and its pollinators, and whether you see, for example, shifts in pollination syndromes associated with change in the available pollinator pool, for example.
So we know very little on many of the Rand Flora genera. That's amazing. From what we know, they have very different pollination syndromes. So some are sunbirds, or we think they are sunbirds. In Africa, in the Canary Islands, they are passerines, or we don't know. But the idea is that, for example, in the Canary Islands, there were relatives to sunbirds and they got extinct. There are actually some fossils. And but we haven't looked into that in detail, in depth, because we know very little about seed dispersal or the pollination. I'm sure there is a component there. I don't think there is a component of coevolution, like the actual pollinators have in a way got extinct, and because of that the plant got extinct in those regions, for example in the Canary Islands. But I'm sure that there, for example, in Canarina, that's one example in which they are pollinated in the Eastern African mountains by some birds, and then in the Canary Islands, as I say, by generalist birds, passerines. Okay. And but we think it was different in the past, for example, like at the end of the Miocene, because there are fossils. So I'm sure there is one component that is not abiotic, that is not only the climate going there. There must be a biotic component, but maybe because the pattern is clade-wide, so it's affecting many different families and genera with different pollination syndromes, I tend to think that is more abiotic. So it's something that is affecting, not simultaneously, but there is, like an umbrella affecting all these groups. If it's biotic only, then you will expect not to see this congruence in patterns. That's what I think.
Yeah, I was thinking more just as a byproduct of the, whatever's causing that sort of biogeographic pattern, that they might be forced to shift pollinators, and that perhaps, I guess, for example, groups that are more labile in their pollination traits that can shift more easily might be more sort of predisposed to being able to bridge these long distances.
That's interesting. And then we will sort of think we'll see a pattern of these Rand Flora groups that are relics. They should have pollinators that are rare now and becoming extinct. So they have no generalists, for example, insects or syrphids going on. We haven't looked into that in detail. There are even lizards in the Canary Islands that act as seed dispersers, not pollinators.
Oh, cool. Yeah. I think the first lizard dispersal system was described recently from South Africa, actually, from the Drakensberg.
Okay.
It's Gunneraceae, I think, some funny thing.
Yeah, in the Canaries is this Galotia, these large lizards, and they are one of the most important seed dispersers.
Oh, cool! Yeah.
But it's something that, you're right, as something that we haven't looked in biogeography. We tend to look at the abiotic, and we don't consider interactions. One aspect I didn't show in my lecture is the idea that you cannot only look at this pattern of factors affecting driving processes. You need to look at interactions between species, and that's a big modulator. And that's something we are actually right now introducing, and it's very much easier to introduce in simulation-based approaches. Interactions. But there are also model-based approaches that have interactions there. Yeah.
I guess do you have decent sort of floras and community level data for that sort of thing? Because then I think there's a publication by Overcast where they do this explicitly looking at that. Yeah.
Yeah,
really cool stuff.
Overcast. Yeah, he has this, I don't remember now the name. He has this simulation approach that is both micro and micro.
I think it's called MESS or something.
Exactly, MESS. That's the. And it's a nice bridge between also ecology and evolution.
Yeah.
So this is one of the ways forward, to have those models on building up from genetic divergence within populations, to populations, species, community level, and then looking at species richness, abundance within a species, and that's a very interesting approach.
Yeah.
Just a thought about the pollination. I mean, to what extent is there selfing happening in Rand Flora taxa? Is there any information on that?
About what? Sorry, Nicola?
About selfing.
Oh, right. There is very little information about that. I think most of them, they are allogamous plants. They are not, but as I said, at least for the species not in the Eastern African mountains, but in Ethiopia, Somalia, there is even little information on that, as for example on the possibility of hybridization, or there has been introgression. Yeah, about the reproductive system, very little is known. We know more, for example, Euphorbia balsamifera, and those that are present in the Canary Islands and are present, for example, in Southern Arabia and Southern Africa. So I think Cantoloma is allogamous. We don't know anything about Cantoloma rotundifolium, what is the disperser or the pollinator. We think it's again some birds in the case of the Cantoloma liberiflorum in Eastern Africa. In Gran Canaria, we don't know. They live on the cliffs. Maybe even Galotia is working there as a disperser. But it's some, yeah. Need to get a student to work on that.
Yeah, several.
Several students.
Such nice natural history work, especially if you've got genetics to underpin it.
Yes, exactly.
Yeah, yeah.
And this mixture of biotic and abiotic, that's very interesting. How do they modulate each other and which level do they work? Because I guess biotic works very much at with you were discussing before, now this interface of the species, speciation.
Fantastic. Okay, well, I think it's probably time to wrap up. Thanks everyone for your attention, and again, a huge thank you to all three of the speakers. Thank you for your very, very interesting talks and your time. And then I just want to mention the next webinar. You've all been registered, you'll get notices in time for the next webinar. And then, of course, for those attending Isabel's workshop tomorrow, so the link will be open beforehand, at least half an hour beforehand. If you want to join in, Isabel will maybe join at 1:45 and just get things sorted before going on with the session.
Yeah, even half an hour if needed. I don't know how many problems the students have with downloading and installing and running the analysis.
Yeah, maybe a bit of extra time won't hurt. Okay. So then again, thanks very much on behalf of the Society, and thanks again to the FABI team for logistics.